good morning, everybody. So I was asked by Sarah to give this introduction to AI. So I thought that Sergio, who is the expert in the group on that, he is a mathematician and a statistician and applying AI models in the pancreatic cancer projects that we run in the group, he could do much better than myself. So we decided to share this. I will introduce first AI and its application, and he then will comment about the technologies and the technicalities of we are applying. AI actually is revolutionizing medicine and patient healthcare. So from its beginning in 1950s, when it was coined the name of AI, the progression, the growth has been stratospheric with some winters, AI winters, and depressing times that have been occurred because of the lack of computing power during these times and lack of investment during the winters, but overall you can see that the progression has been exponential. There was in the 80s, there was the appearance of new branches of AI, that is the machine learning in the 80s, and 2011, the deep learning of AI. Today, there's many other branches, natural language processing, ambient AIs, and others, generative AI, for example. And well, for example, in last year, there's applications already, and last year FDA approved 691 applications for AI. So again, this is amazing how this field is being changing the way the patients are being cared. So among the applications is cancer, and just to show you that this attracts the attention not only by scientists but also by journals. This is a cross-journal collection asking for papers on covering advances and development of applications of AI in the field of cancer. So in the outline, we decided first to cover the definitions of the different terminologies and concepts of AI, the application, the pros and cons, the input of the data that, AI needs a lot of data, reliable data, so which type of data, including also images and the tools that AI is applying. Then we will show you some examples that we are running in the group, also how we generate synthetic pathological images based on RNA-seq data, amazing world. And then what is of interest to us, that is to apply AI to integrate different featured liquid biopsy features to build this robust composite algorithm for early detection pancreatic cancer. And conclusions for sure, and future perspective that I think it's also quite interesting to go through. So regarding definitions of AI, artificial intelligence is a computer system that can perform tasks that normally require human intelligence. So the main objectives of AI is problem solving and learning. And there's other aspects on visual perception, for example, speech recognition, decision making, and etc. So what it does is mimics human intelligence. Okay, and this was possible because of these technological revolutions in the last half of the last century. Okay, and it has permeated other areas. For example, the image area. For example, with computer vision. So AI is used to analyze images, to report on images, and to interpret images. But as I said, there's other more recent branches like machine learning and deep learning that you probably have heard about that, and sometimes we confuse and we use them, not in an appropriate way. So machine learning and deep learning, what they do is that they mimics even more the way we humans think. The difference is that machine learning applies AI technology, AI systems to allow to learn automatically, to solve problems through identification, classification, or prediction using regression algorithms or decision trees. While deep learning do the same, is within the umbrella of machine learning, but they use neural networks to do that. That it's even a step forward in this way to mimic our intelligence. Okay, so it's deep learning because there are several layers of neurons that it usually apply. So other branches that are even more recent are generative AI, and as I said, Sergio will comment on that, is to generate, based on real data, to generate fake data, new data, synthetic data, or images. And this is, the potential of that is huge, I think, in the future. Ambience clinical intelligence that I will now comment, and a very hot topic that it's natural language processing. And this is how more or less we combined all of these different fields. So ambience AI actually is a system that passively monitor the environment in order to create documents, reports, analysis, or alerts. So in a way, what it does is to take, for example, Alexa, that you may know about that, no, from Amazon, etc. So it takes information from the ambience, and it analyzes this information, and it could provide a predictive alert to, for example, to old people at home, or to decrease physician burnout, or in the hospital journey, both in the urgency and the ECU, or in the operating rooms, some notes and information on that. So this is a new field that is being developed and probably will be applied in our homes and also in our professional settings. And this is a very, as I mentioned, a very hot field that most of the top centers using that are in the upfront of AI, they are starting exploring. And what it does is to allow the systems to interpret the natural language, our language, written or even verbally language. And for example, this is a way that it could be applied, for example, to explore in the e-medical records, electronic medical records, that they are not structured, they are unstructured information, the information that we are interested in taking, in recovering, for example, on family history of PDAC. In this huge amount of data that 99.99% is non-informative. So these systems does allow to recover the important information that in a big lake of noisy information. So applications of AI are many: telemedicine, remote monitoring, diagnosis and detection. That is what we are interested in PANCAID. Drug discovery and development is amazing how the structural biology, they use AI to assess the ligands to the molecule, which one fits better with the molecule of interest, the mutation gene, for example, and how do they do in a super rapid way. Treatment planning and personalization, prediction analysis and risk assessment, and also managing of the patients. The pros or just the following of the previous slide. So of AI, you can guess about them. It's a enhanced diagnostic accuracy to define the conditions earlier, PANCAID, for example, and more precisely, we hope so, to enhance predictive analytics, to identify trends, forecast disease progression, facilitate early interventions, personalized treatment by integrating different layers and modalities of information, and also considering here image data management and even patient engagement and educational in the clinical trials or in the studies or in the treatment. The hurdles of AI, there's many here. And what we can identify among them is the data privacy and security risks, and you can also guess about that. There's an issue regarding bias and fairness, regarding how the models have been trained with non-generalized data versus generalized data. There's regulatory and legal challenges. There's resistance to adopt the algorithms that have been developed or are being developed. There's ethical concerns, data quality issues, and cyber security risks. And to finish on that, you may heard about the debate that is being now placed in UK regarding the AI plan, and probably you can comment on that further. That the AI plan, this is what is my understanding, have considered to share data or to sell data to private companies in order to speed up the development of algorithms and devices, but this has provoked an important debate, and probably this will be also expanded to the rest of Europe. No? So national health systems may face this kind of data. So with that, I leave now Sergio.
Yeah, thank you very much, Núria. So it's a real pleasure to me have the opportunity to present here, and I'm going to do an overview of some of the AI tools that are most widely used in the practice. And also, of course, I will introduce the input data, which is truly important because this type of AI algorithms works totally depend about the quantity as well as the quality of the input data that we are going to use. Yeah, so to create a robust model of AI, of course, we need data, because this model works better when we have more data available. So here, we can distinguish between omics data and omics data. In the group, we use both. For example, in omics data, we have genomics, epigenomics, transcriptomics, proteomics, metabolomics, plenty of different data types, and also we integrate all of them with non-omics data, clinical pathological, demographical, epidemiological, a lot of data. And also, depending on when we use this data, when we integrate this data, we are talking about different techniques of AI. If we use only one model that uses a single layer, we can talk about independent modeling. If we use different models with different layers for the same outcome. Also, we have conditional modeling if we integrate another omic data here in the middle of the step, or one step of the process, or joint modeling if we use, at the very beginning, we combine both and we try to predict an outcome. One that is very important, and now AI is revolutionizing all this field, is medical imaging, and we are working very hard with medical imaging. And as you know, medical imaging refers to the set of techniques and process to create image of the human body or parts of it for clinical purposes, like diagnosis, examination, prognosis, treatment effect. Depending on how these images are generated, we can talk about, for example, CTs, MRIs, ultrasound imaging, and digital pathological slide. Here in the group, with Núria and the CNIO, we work with radiological images and also with digital pathological slide. So without going into details, the medical imaging is very important because it's present during the whole process, from the detection and early diagnosis. Also, it's very useful to decide with CTs and MRIs in some of the cases if a patient undergo to surgery or not. Also to define the staging, confirm if it's, for example, a PDAC or it's a CNR cell, also the treatment response monitoring. So it's present during the whole process. So if we are able to extract some features from the image and we can create models that take into account these features, maybe we are able to generate models, complex models that can identify some characteristics that are cannot identify by other type of input data. How we can extract these features from the image with deep learning. Núria already mentioned. I'm not going to go into the details here later because it's plenty of time, but basically the most typical use model is the convolutional neural network, and this model allows us to convert an image to a numerical vector. So for example, here in reality, an image is a set of pixels, and a pixel is a value and a number between 0 and 255. So if we think an image like a matrix of numbers, we can, with matrix operations, we can reduce and convert this image to a numerical vector, reducing the dimension. Or, for example, a probability. I can convert this image to a number that is saying me the probability that that image contains or not a tumor, for example. So this is possible thanks to AI and thanks to deep learning. Also, we can try to relate an image with another image, not with a vector, with another image. And I will comment on that later. And depending on if we focus on radiological or pathological, we talk about radiomics and pathomics. So radiomics refers to techniques used for the quantification and stratification of this type of radiological images. And this is the basic overflow of radiomics. Of course, the first step is acquire the image. That depends on the machine. And then we move to one of the most important and crucial steps of radiomics, that is the tumor labeling, the tumor segmentation, where delineate, annotate, or we paint our region of interest. That can be done manually and automatically. I will explain later a little bit, but this is a crucial step. And then once we have our region of interest, we extract the features. And to do that, we use here deep learning. But at the very beginning, it's also possible to apply some machine learning techniques that can extract features more related with the shape, with the histogram, with the texture of the colors. Sometimes we can apply also this type of techniques. And when we have the features, we relate that features with an outcome and do some statistical analysis. With pathomics is the same, but we change the radiological by the whole slide image. The only step that changed a little bit is the image preprocessing, because now we are working with two-dimension images, and in the radiological are 3D. And here to do the preprocessing, sometimes we need to remove the white background that is behind. We need to normalize the color because the staging is sometimes different depending on how it's generated. And also, these are very heavy images, and we divide the whole slide images into smaller images that are called tiles or patches. But at the end, when we have do the preprocessing, we extract again the features, and normally that's used with deep learning. One of the most crucial steps is define our region of interest, what we want to extract. So here it's the tumor annotation, the segmentation, and we want to delineate our region of interest. The thing is, delineate of the region of interest is usually done manually, and we need to change that because it's very time consuming. It requires a lot of effort and introduce variability, the human variability. So for this reason, the neural networks can allow us also to do this segmentation automatically. And the most well-known model here is the NNUNet that performs very well. For example, when we try to segment automatically the pancreas in our case with accuracy about 80%, but in the tumor the values are, the top one is 61%. That is not enough. So we are trying to improve this methodology because we need this automatic segmentation to reduce this variability, reduce the times, and that's very important. And this is done again with CNNs, with convolutional neural networks. Once we have seen more or less the AI in general, the image in particular, I want to also introduce some examples that we have done in our work. The first one that I want to introduce you is using radiomics indeed. And this is in collaboration with Nana Xue, that he's a PhD in our group, and we did like a model, it's called PMPD, that it's able, using the CT image at diagnosis time, is able to predict if a patient has organ metastasis in any organ at diagnosis time using only this CT image. Another example that I would like to show you is a model where we integrate a lot of different omics layers to predict prognosis of the PDAC patients that we have done with Evangelina and Lola in our group. And just to finish, I would like to introduce you this novel field that Núria have already commented, that is the generation of synthetic data using AI. So this giving us another opportunity to create different models that integrates more data and increase the sample size in some of the projects. And to do that, we are using AI, and more specifically, diffusion models that are able to generate data similar to the data which they are trained, and works destroying data with a successive addition of Gaussian noise. So it's like a sequential step, and it's adding noise in each step, as you can see in this image, and it's learning how to recover the previous image. So without going into the details, for example, this kind of DALI that is included in ChatGPT works like with these diffusion models. There are plenty of studies that are starting with use these diffusion models. And this one, for example, that is very interesting because it's not generating image from the noise, it's generating image from the RNA profiles. And yeah, here you can see, for example, in this principal component analysis, how the model is able to recognize and stratifies different characteristics of different cancer types, lung, glioblastoma, different ones. But also, if we focus on a specific cancer type, we can compare between real and synthetic data, and the cell distribution is similar. This is a very promising results. So we decide to adjust this model in a way and create our own image generation, synthetic image generation, but focus just in PDAC. We change a little bit the data preprocessing, the model, but no worries on that. These are like our preliminary results, and here you have two sets of images. One are fake and what is real. And yeah, for the vast majority of the pathological experts are even undetectable which one are real or synthetic. So we are very promising with these results, and for sure we will allow us to use this information in future projects, in future models, and more or less that's all I want to comment.
Thank you, Sergio. So Sergio has shown you how by integrating multimodal data, we are able to improve the predictive models. In this regard, regarding overall survival, but what we want in PANCAID is to predict the presence of the tumor itself. So the pancreatic cancer tumor. So this is the aim of PANCAID. What we want here is to integrate different liquid biopsy features that has been proposed to do in different steps in the disease process. And to do that, AI, mainly machine learning, we plan to apply machine learning, the Bayesian kernel-based model that Sergio is commenting. So this requires a lot of data and reliable data to get the model, a robust model, but this is a long way, and you all know that this is a long way to get the data, to build the model, and to validate internally the model. But when we have the model, this is not the end of the process, because after that we have an evaluation step that requires external validation, requires updating during this period, and also to impact the assessment. Also considering cost-effective, Hans Helmut is here and he is involved on that. So there's different steps that actually the project also consider here. So these integrative efforts poses several challenges, quite a lot of challenges altogether. I want to emphasize these three that regards to the lack of studies to validate and replicate results. For example, in PANCAID, having external studies that have all of this huge amount of data and different modalities of data will be very difficult to have access to them in order to validate them. Let's see. There's also the interpretability of the study, as Sergio has mentioned, at the biological level and also at the clinical level. So this is a very important step, and also to consider the dynamic nature of the process, because maybe the composite algorithm considers and integrates one type of features when it's 1 year before the diagnosis of pancreatic cancer, but different features when it's 5 years before. So it depends on the time of the process that the algorithm may be different. So we have to consider all of these challenges. To conclude on that, well, AI is rapidly transforming medicine, and in pancreatic cancer, the results are highly promising, and we should not give up and to boost. This is difficult to work on pancreatic cancer research, we all know, but this is super important, so we have to boost and enhance this research. Natural language processes and ambient AI is transforming the field, and probably they can also apply to also monitor the patients and in healthcare. In pancreatic cancer, medical imaging, no need to emphasize the importance of combining and integrating these kind of different modalities of data. synthetic data and images may help in order to boost the training models. So the need to develop automated workflows and algorithms to monitor and to identify earlier the pancreatic cancer. And for that, we need data, reliable and harmonized data, clinical, omics data, super important, and then to build the model, to evaluate it, to interpret, and to impact the assessment. I want to finish to show you what I thought would be the future, but actually it's the present. And actually, this will be the devices, the technology that we will actually has been already proposed to have. So these technologies that combine different types of data modality, from liquid biopsy to image and to other type of data. There's already a couple even psychological barriers. So I don't know how to measure that using the VC, but it's not only one paper, but there's other papers that have even assessed the impact of this kind of technology. And they use images, for example. But in any case, just to show you that AI is revolutionizing medicine and healthcare, and we are there. So next step of PANCAID probably is to think on other steps. No? And with that, to thank you all for your attention, and to the members of the group that have been mostly involved in what Sergio has presented. So Lola, Evangelina, Mireia is here, Adrián, Brun, Pablo is here also, and Sergio. Thank you very much.